Yes, good afternoon, everyone. We will start discussing cDNA library. So in today's class, we will talk about all cDNA library, and it is going to be about uh, 5 or so of methods. It'll be a good idea if you can note down so that and try to understand the key points. But otherwise, the video will also be there. But it will be because it's 5 or so, you might have a question of confusion. That's why. So what is cDNA? It is complementary DNA, right? Complementary to the mRNA. That's the point of that is cDNA. And it's usually what we say is it should be in double stranded. Many a times in our purposes, we use single strand too, but also mostly for cloning purpose, as you must be aware that it's in when you want to make a recombinant DNA of the cDNA, it should be double stranded. The ones that we use is the spliced mRNA, because when mRNA is produced, it is produced as a pre-mRNA, right? It has exons, it has introns, and so on. And you will have splicing happening. These are the shaded ones are introns, and this in the nucleus will get spliced. I mean, spliced in the sense this is exon 1, exon 2, and exon 3. The these 3 exons will fuse. I mean, splicing means attachment or joining of exons. So you will get a mRNA that is finally with the mature mRNA. It has the 3 exons like this. And if you remember transcription, you'd also note that there is a G cap on the 5' end. It's I think I encourage you to try to find out the chemical structure of the G cap. And there is also another part is that there is a poly tail. These are features of eukaryotic mRNAs. Okay, one second. Okay, yes. And alternate splicing is one more thing that I would want you to find out. I mean, these are some things that are important that you have to remember. Alternate splicing also. So the first thing, when we have steps, we have several steps. First one would be to isolate the mRNA. Okay, we first have to get the mRNA, that is isolate the mRNA, and then use it as a template to synthesize cDNA. I think these protocols are pretty straightforward. It is using oligo-DT. As I was mentioning before, all eukaryotic mRNAs, they have a poly A tail. So you will have, there will be other RNAs like tRNA, rRNA, and other small coding RNAs also. And you will also have mRNAs. mRNAs, as I described in the previous slide, they have these, a poly A tail like that. Okay, like this. So how can we separate these RNAs from mRNA? That's the question. So using the feature of this poly A tail, we can do that. So if we take oligo-DT beads, that is, you have beads that are attached to oligo-DT. We are taking it oligo-DT because it would have been then complementary to the poly A tail. These are the other non-components, like I described in this case, okay, which are not mRNAs. These are mRNAs, and the other ones are non-mRNA RNAs. So the oligo-DT containing beads will base pair with the poly A tail like this, and the rest of the components do not stick and they will wash out. And then we will elute using changing the salt concentration or alkaline conditions. No, we don't use alkaline conditions for a reason. By changing pH, by changing salt concentrations and the temperature also you can use. You can wash off the mRNAs. Now you have RNAs that are that are devoid of other mRNAs. Okay. So step goes that you will first isolate total RNA. Total RNA consists of both mRNA and non-coding RNAs. So using oligo-DT, you will have isolated the RNA like this. And then from here, you would try to synthesize cDNA. And once the cDNA is synthesized, you would have constructed a recombinant or a clone. Okay. So from here to here, getting to here, we have some issues and we have some solutions too. Based on our objectives, we have several ways of how things can be done. That is what we will see. We will first look at very primitive methods. And this is the one, it's a primitive method where this is the RNA. Always, as I mentioned, you should always indicate the 5' and 3' end. Okay. So a general strategy most of you must have learned already is that because you have poly A tail, then you can use a oligo DT primer, which would have 5' here and 3' here. Then you will add a reverse transcriptase enzyme, and then this would synthesize the first cDNA strand like this. Okay, like is shown here. Occasionally, what may also happen is that this is the synthesis of first cDNA strand. Okay, first cDNA strand. Yeah, we have to be specific. First cDNA strand, and the 3' and at the end, sometimes it forms a hairpin loop like this. So if we just add DNA polymerase now, it can start synthesizing the second one. But then what is obstructing with the rest of the first cDNA strand? You have RNA is still base paired like this. And if you want to get rid of that base pairing, one should have, if you want to remove the RNA, you can use alkaline conditions. Why are we using alkaline conditions? Anybody? What happens if you add alkaline conditions, and why RNA or mRNA degrades with alkaline conditions? Anyone? Quickly, please. Yes? So I'm trying to draw the ribose here, right? And say here is the glycosidic bond with that of a base. The pen is not writing well, I guess. Okay, and then you have first carbon. This is second. I should put prime because that is what we are indicating. This is 3', and this is 4', and this is the 5th prime. CH2 OH, something like that. And here would be the phosphate from the other ribose, something like this. Right? This would have been the other base. So again, you will have 1 second, 1 prime. This is the 2nd prime, 3rd prime, 4th, and 5th. With the 5th phosphate group is there, 5' phosphate, which is now which is a type phosphodiester bond with the previous ribose. So what is the difference between DNA and RNA? RNA has ribose and DNA has deoxyribose. And what is the chemical difference? The difference is that RNA ribose is this 2' OH. Okay, assume that this one would have been going this strand would have been like this. I'm just discussing here. When you put in alkaline conditions, the H plus that is present here is removed. So this O would form an oxyanion, which is highly negative, and which will make a nucleophilic attack with the phosphate backbone here, because of which this bond here is broken. Okay, I hope you also get the terminology right. What is denaturation? What is degradation? Denaturation is if two strands are there, base paired, and they become single stranded like this, then it is denaturation. If you have the same nucleotide or DNA or RNA, it has broken into pieces like this, that is degradation. Okay, denaturation is kind of reversible, but degradation is not. Anyway, so the 2' OH of the ribose or the oxyanion that forms in the alkaline conditions will make a nucleophilic attack on the phosphate and results in degradation. Okay, that is why we are using NaOH. So now we got rid of the mRNA. All we have is first cDNA strand. Upon addition of DNA polymerase 1, it will synthesize the second strand as well. The question is it second strand at all? Is it one strand or two strands? It's actually a single strand, right? It is continuous phosphate backbone. But we can make it by using S1 nuclease, which is a single strand specific DNAs. So usually this end, although it's shown like this, would be something like with a small hairpin loop like this. So in all of this DNA, if you see in all of it, only this part would have been the single stranded one. So S1 nuclease would degrade this DNA like this. And now what you will be left with is a double stranded cDNA. Okay. As we keep looking at each of these protocols, we will have several advantages and disadvantages. Try to keep questioning on themselves, what is the disadvantage of this, and so on. Okay. I hope the objective should be very clear. What is the objective? We want to clone the complementary DNA of that mRNA. And every polymerization that happened by reverse transcriptase is the information that is present on RNA. But we are using S1 nuclease here and degrading some part of it. That means we are actually losing some information. It is incomplete on the 5' side. Okay, because we are using S1 nuclease. That is the disadvantage of this particular protocol. There are also, this is a little more exhaustive one, which employs the same method where we use first cDNA strand synthesis using reverse transcriptase, then treating with alkaline conditions to get rid of RNA. And then we will synthesize the second cDNA strand like in the previous what I have discussed. And also they use the same S1 nuclease. And S1 nuclease will create the double-stranded cDNA. Sorry, pen is a little bit troubling, but yeah. Double-strand cDNA. And now to clone it into a vector, we could use homopolymer tailing. I hope you remember that. Terminal transferase is added and DGTP is added here so that you will have on the 3' overhang that is generated by PST1. Multiple Gs are added on both the strands at the 3' ends. And similarly, the cDNA is treated with terminal transferase and DCTP. And therefore, you will have multiple Cs and Cs based on the multiple Cs on the 3' ends. Although it is shown like this, it is implied it is 3'. I hope you can see it here also. That is the 3' one. Okay. just for ease. Then you would have added the uh, you would have mixed these two because you have multiple G and C's. G and C has 3 hydrogen bonds, so you will have more number of hydrogen bonds to hold them together. Then you will transform it into cells, which the host repairs the gaps and then you will have your clones there. Okay, so this is the very earliest method, which is predominantly using the key is or the key feature in this one is using reverse transcriptase followed by S1 nuclease. Cloning is simple using homopolymer tailing. If I were to make a notes, this is how I would do as well. RT S1 nuclease homopolymer tailing and then transformation. When you are supposed to look at something, this should be your notes so that you can always recall. Just before exam, if you have to see, if I'm just looking at this, I should be able to find out. So at this time, your understanding of what each one means, you should know. And before exam notes should be something like this. Imagine how many protocols you would actually remember just because you wrote something like this in one single page. You can do that. Now there is a disadvantage. Another disadvantage that I didn't speak of is we are not sure if the cloning in homopolymer tailing will be directional. Is it start codon to stop codon, or is it the reverse? Okay. If this is not an expression vector, we should have cut this out and cloned it into an expression vector and then further analyzed. But as such, it doesn't seem like it is directional clone. So we need to improve a directional cloning. I like this method for one of the reasons. I also asked you a similar question, if not exactly the same. Everything is almost same. Reverse transcriptase, alkali, and then we have DNA polymerase. I hope you remember what is clinofragment. And now here comes something. And the objective you should assume is that this is the promoter, and we have to clone in this direction, right? This is the start codon, ATG here on this side, and then stop codon is somewhere here. So what we will do is, excuse me. Yeah. So if you want to make it directional, you should have one kind of linkers on this side, another kind of linkers on this side, like this, separate ones. So what this is a smart one, a smart way of doing it, because I like it. Here, prior to cleavage with S1 nuclease, they have added the SAL1 linkers. Okay, they have added SAL1 linkers. Why did they add prior to S1? Because now the only place where SAL1 can actually bind or ligate would be on this side here. It cannot ligate on this side because it is continuous. There is no substrate for a ligase to act upon. And then after that, they treat it with S1 nuclease. And now you have a double stranded, say, blunt end here. And now you can add your eco R1 linkers, something like this, which will add here as well as here. It doesn't matter if it is added here or not, because once you cut with SAL1, although there are multimers of linkers here, this one will be cut like this one also. But eco R1 or SAL1, they can cut everywhere. But what we are going to take is only the fragment that is useful to us. That is what we are going to use it as a fragment for ligation to occur. So if we have a vector must be having eco R1 on this side and SAL1 like this, and then we would have cloned this or ligated the fragment with in this way, and always it would have ligated in the improper orientation. So upon expression, it will be able to produce RNA and the appropriate protein also. Okay, so for directional cloning, these people have done this one is a smart step, although I don't think it is of a great technique, but it's a good thinking, and that is why I like this one. I also asked you in your second assignment, how would you add two different adapters to onto two different sites? I hope each of you have spent enough time trying to think of ways. It is about how much you think. It's not about knowing. It is about thinking, right? So there are further improved methods. This is also in terms of directional cloning you can think of, or full length. Here there is one problem. In the previous one, all the techniques, three techniques we have discussed before, in all of the cases we are using S1 nuclease. If we are using S1 nuclease, it means that we are losing some information here, and that's actually incomplete mRNA, and that's not the we don't it's insufficient to us. So how not to what can we do if we don't want to use S1 nuclease? That's the question here. So mRNA and then reverse transcriptase is added, and the first cDNA strand is synthesized. Okay, now they have done something here also. You have something interesting, or the thought process is good. They added terminal transferase already to this. So terminal transferase would add CCC here, and also here to the mRNA as well. It doesn't matter as long as it has a accessible 3' OH. And once it adds, then you're treating with alkaline conditions, because of which all the mRNA will be gone, and this is also gone along with the mRNA. It's not going to, because once you recover full length, you will get rid of everything else. Now you have something, the first cDNA strand. On one side you have poly T, on the other side you have poly C. Because you know this is the poly C, you can use a primer that is poly G or oligo G, GDT, DG. And then using that primer, one can synthesize the second cDNA strand. And have we lost any information? We did not lose any information from this side, because we did not use S1 nuclease. No degradation was made. This is likely to allow us to clone full lengths without any degradation. Okay. So quickly in this method, what they did is to avoid using S1 nuclease for the synthesis of second strand. But then it is very difficult to know what this sequence is. So what they have done is they've added terminal transferase, because of which and DCTP. So now we have C. Previously we didn't know what sequence is here, but now after treating with terminal transferase, we know what sequence is here. Because we know that it is poly C, we can add a poly G primer and allow the synthesis of the second cDNA strand. Okay. So I hope this is the same thing as what we have seen before. We don't have to repeat. In this case, we have directional cloning. And in this case, we have full length. Here, the thought process is about how to add two different linkers on either side. And in this protocol, in the one here, it is about, if we are not using S1 nuclease, how can we clone the second cDNA strand? And this one is about using terminal transferase, so that we know the information that is on this end. Then we could have used a primer such as poly G, oligo G, and then synthesize the second cDNA strand. Okay. Although we say many of these are advantageous and so on, there are complications to this, because of which we do have other uh, there will be some problems associated with it. We will come to that in a while. This is one other protocol. You want full length, okay? The objective is full length. That means we should not use S1 nuclease. Second thing is we want it directional. Okay, those are the two objectives that we have for this protocol. So here, the first rDNA strand is always the same in a majority, in all of these cases. It's always reverse transcriptase and polymerization. And the one thing that is different here is that one is using a oligo T with a primer of Sal1. Okay, I hope you remember when we designed cloning primers, we had 3 parts to it, right? Um, One part is the part. This is the part that will bind to the something is. This will be the part that is binding to the or homologous to the sequence of amplification, and then we have a restriction site here in this place, and then we had a buffer sequence to allow increased degradation by restriction endonucleases. In a similar way, what we are doing here is we have oligo T, and then we have the Sal1 primer attached to it. So when the first cDNA strand is made, it already has the Sal1 linker here. And now for the second cDNA strand, the RNA removal, everything is alkaline based, and that is normal. Now for the second strand, we are using DNA polymerase. And what we are using here is random primers. Okay. I want you to find out what are random primers. Pen is not writing, but we will adjust it. This is random primer, okay? They are small sequences, maybe 6 to 10 bases. You'll use it such a way that the frequency of it is high. So if you have a DNA like this, and random primers can bind, they are small nucleotides. It could sometimes be the same type of nucleotide. Sometimes people use a mixture of each one, may be some different sequence. So you can have a mixture of sequences or so. So the objective is that you want it to be each RNA to be bound like this by one or more primers. So the random primers, they can bind like this, and all you need for a polymerase is a primer template junction. And these primers are going to provide that primer template junctions like this. And now you are again using Clino fragment. That means it will identify this and will synthesize the DNA and stop. Well, from here you have, because it does not have, Clino fragment does not have 5' to 3' exonuclease activity. So it does not cleave this part. It will rather simply, it will synthesize the next part like this. So multiple polymerases can bind here and synthesize these fragments. That is how you will get a mRNA that is likely to be full length. Okay. If we want to argue that this end, if it doesn't bind, what will happen? In that case, you may not have. But as I said, random primers are chosen such a way that you increase the probability that they would bind to most sequences. Or it could be a mixture of sequences also. So second cDNA strand is synthesized using random primer. Now it is simple because here we had Sal1 primer like this one, right? We already incorporated within the primer like this. So the first cDNA strand would have the Sal1 linker. When the second cDNA strand is synthesized in this way, Sal1 is also synthesized. So now you have a double-stranded Sal1. Here it was only a part of single strand, right? And now you can add EcoR1 linkers. When you add EcoR1 linkers, it will add here and also here. But it doesn't matter, like we have seen in the previous protocol. Once we use SAL1 and EcoR1 restriction endonucleases, SAL1 will cut here, EcoR1 will cut here. And then we can just clone it into a vector that has the proper orientation. So it should be something like EcoR1 or SAL1. So my pen is little, yeah, something like this. So what are the advantages of this? In the same protocol, we can generate a full length cDNA, and we can also make it a directional cloning. Those are the advantages of this. Quickly, the key points in this protocol is adding the SAL1 primer to the SAL1 sequence to the primer. First, oligo DT primer. Second thing is using a random primer to generate the second cDNA strand. And then the third one is to add the linker, two different, I mean, one is already added, SAL1. Now you add the other one, other linker, EcoR1, and then digest and clone. Okay. So there are several limitations to the conventional methods. They could be, if we have a cDNA that is very long, then there are several possibilities that the reverse transcriptase that is transcribing, say this is the 5' and this is the 3'. So the primer is oligo DT is here, and reverse transcriptase is synthesizing. Because if the sequence is, if the template is very large, then sometimes the reverse transcriptase may not be, the probability of reverse transcriptase falling off is high. That means you're actually having incomplete or shortened fragments, incomplete cDNAs. That is one of the problems. There is one more problem. You may also have RNA, mRNAs. Some of them are full lengths. The same mRNA, if it were broken, so in some cases, because of some reason, the mRNA might be broken, the same one. Actual length should have been this much, but it is broken. And that is also a problem. Okay, sorry. The broken one usually could be something like this. Sorry. Say you have poly A tail here, and this part is broken. So this will be the new 5', and this is the 3', and this would have been the 5', right? So there are two problems that are that we are coming across. First one is that reverse transcriptase may not synthesize the full length of the large genes. Second point is sometimes there are broken mRNAs within the sample that you made, or it might be in the process of degradation too. What to do now is the question. How can we get full length cDNAs? First one, I think we'll only discuss last two. I'll stop with that. And sorry. So the first one is called capture method. It is, name itself implies that we are going to use the GCAP. We are basing this on GCAP. We are going to use two things. First thing we are going to use is D oligo DT, right? This is our primer. That means we are targeting the 3' end, right? Sorry, 3' end. What we are unsure of is the 5' end. What does the mRNA have? On 3', it has a poly A tail, and that's why we are using the oligo DT primer. And on the 5' end of it, it has a GCAP, right? Okay, I'm unable to write. So G cap. Again, simply I will put it G is like this. It is for protection, and it is also important for recognition during translation. So it is a unique kind of phosphate bonding. We need to also read it. Some other time we'll do that. I hope I'll be able to solve these problems by the next time. Let's check. So there is a phosphate, another one, and then you have the mRNA and AA like this. Okay. So the G is coupled, then you have three phosphates, and then you have the mRNA starting sequence. Okay. So I hope you remember, you understand that it is not vulnerable to degradation directly. So the first cDNA strand, assume that there are two copies. One second, I will erase these and then I'll get back. So now I'm going to do the same. I have a full length and I have a truncated one. I also have the part of it. So this would have been the full length 5' to 3'. So on the 3' you have oligo A or poly A tail. On the left side you have the full length 5' to 3', which is having the poly A tail. Now I'm going to use oligo T and try to amplify it. This is oligo T and here is oligo T. And when I add reverse transcriptase, this will synthesize until here. This is broken, so this becomes irrelevant. This fragment becomes irrelevant. Only those that have poly A tails are actually going to be polymerized or reverse transcribed. That's the correct one. And here, for the full length, it will reverse transcribe until the end of the mRNA, right? Now, if we treat it with, sorry, this is for a different purpose, right? I told you two different purposes. One is for full length, one is for broken mRNAs, and the second one is for incomplete reverse transcription. Okay, I'm sorry. Here is the full length. This is about incomplete reverse transcription by reverse transcriptase. So if the reverse transcriptase has transcribed till here and did not transcribe this part. So what happens? How can we remove these and select these? That's the question. Sorry, a correction there. So here you have GCAP, and this part of it is not transcribed, as I'm showing here. This is not transcribed. So if you add RNase A, which is a single-strand RNase enzyme, it will break off this part that is not base paired with this one, with this partially synthesized strand. In here, it will not degrade anything because it has transcribed. Reverse transcriptase has transcribed this part. So this is important. You have a column to which you may have, sorry, no. So we have a column, and the column has to the attached stationary phase, you can assume that we have EIF4E. During eukaryotic translation, this is the initiation factor in eukaryotes. That is the one that recognizes the GCAP. Okay, that binds to the GCAP, and then the pre-initiation complex is organized and translation happens. So you're using it as it's something like a ligand and binding factor, right? Here the ligand is something like the GCAP. Now if you allow these two to pass through, now the one with the GCAP, here is the GCAP. In this one, the GCAP is no longer present or available. It is broken down, or because because we treated with RNase A. Here also we added RNase A to the mixture, but the full length one will be protected because reverse transcription is complete. So the mRNAs that have the GCAP will bind here along with their first cDNA strands. The rest, like these ones, it is reverse transcribed this part, but it does not have GCAP, so it is not retained in the column and will flow through. Okay, and now after washing the column, we will elute this and process the second strand synthesis in methods that we have described earlier. Okay, so let me say it once more before I go to the next slide, last slide. Here in capture method, as the name indicates, it is about exploiting the presence of GCAP on eukaryotic mRNAs. So we can always find out the 3' because majority of the eukaryotic mRNAs have poly A tail, and the 5' on the 5' end they have GCAP. So this is about as a solution to solve those that do not, in cases where partial reverse transcription happens. Okay, so the partial reverse transcription, something like this happened, and if you treat with RNase, this part will get degraded like this and will not have the GCAP anymore. These partially reverse transcribed fragments do not have GCAP, whereas the full length ones will retain their GCAP. So when you process it through a column containing EIF4 4E transcription factor, those with because it is specific to GCAP, all those that have the GCAP will bind and the rest will flow through. Once after washing, we can elute the full length cDNA, then synthesize the second strand cDNA, and then clone it or ligate it into a vector as per our need. Okay. So in the next one, this is the last. Here the objective is what I was trying to say in the beginning: how to get rid of mRNAs that are incomplete or broken mRNAs. So here is the full length, and you may also have other ones. Where it might have been broken here. Okay, assume that this is a discontinuity here. We have poly A tail and poly A tail. We do not want these kinds of things in the library. If I forgot to mention, I think I will continue a little bit in the next class about cDNA libraries, because we need to discuss about screening. When I say library, that means we are making library or cDNA of all the mRNAs expressed in that cell at that time in those conditions. Okay, it's not one gene, one mRNA. It is multiple mRNAs. So in many cases, you may have broken pieces like this, and we don't want to have anything broken mRNAs like that, because it will complicate our assays. What if we might think it as a new or truncated new gene or something like that? So we want to avoid we want to eliminate these kinds of broken mRNAs from our library. Here is a method that is a little bit complicated, but simple enough. We can always get through it. So there are two different mRNAs here. One is a full length one, and the other one is a broken or partial length one. Okay, what is the difference between these two? They both have poly A tail, and the difference is here. Full length one has the cap, and the partial one does not have a G cap. So they treat it with, now the step is to treat it with bovine alkaline phosphatase. So the alkaline phosphatase will remove the phosphate group that we have learned before. Now, what does it have on the 5' side? It has a OH, and the full length one has a G cap. Alkaline phosphatase cannot act on this because it is a G cap. As I showed you, the structure of G cap is little different. Then they treat it with TAP. It is for acid pyrophosphatase. I don't remember. I can't recall what T is. I think I have to check that out. But the this is the chemical reaction or the enzymatic reaction that it does. Acid pyrophosphatase. As I described before, G, then you have phosphates, three phosphates, and then you have the bases, say GACT, mRNA. Okay, I shouldn't put T there, right? U, A, C, A, like that. This part of it is the mRNA from here. This part, you can think of it as the GCAP. So pyrophosphatase, what does it do? It cleaves the bond here like this. The GCAP is removed here. Okay, after two phosphates, what is left is a 5 prime single phosphate group here. Okay, acid pyrophosphatase removes these two because it is two phosphates along with the G is attached, of course. We call it as acid pyrophosphatase. Now, this is also a nice technique. I like this for the thought. They add oligo along with and use RNA ligase. So if I add a RNA molecule, oligo, I have a small RNA fragment of known sequence. I know the sequence of the RNA, and I add RNA ligase. It's different, similar in action to DNA ligase, except that now it works only on RNA. What will be the substrates for it? 5 prime phosphate. This is 5 prime, and 3 prime of the oligo is like this. It has a OH, and what it needs to make a phosphoester bond would be a 5 prime phosphate. The full length fragment, like here, full length mRNA has a 5 prime phosphate. Why did it have? We have removed the G. It had the GCAP. We removed it using acid pyrophosphatase, and now it has a single 5 prime phosphate group. And the oligo that we are adding is having a it has a 3 prime OH. So a phosphorus bond is made between these, and now you have the oligo capped. The technique name is also based on that, oligo capping. You are adding an oligo to the full length mRNA, whereas the fragmented one, it has a phosphate group, and it cannot base pair or it cannot ligate with the oligo. So only the full length mRNAs will get the oligo cap. And now, because I know the sequence of this oligo, I can use a primer. Okay, you will just synthesize the first cDNA strand using oligo DT, and you will synthesize till here. Because I already know the sequence of this one, I could use this sequence, use this part of the sequence as a primer to synthesize the second cDNA strand. Okay, that's how you can perform or you can synthesize the second cDNA strand much more easily. Okay, I'll stop here, and you can ask me any question. Before that, I will summarize this once again. The objective of this oligo capping method is to get rid of all the broken mRNAs. We don't want these. So the treatment goes with alkaline phosphatase to remove the phosphate group because only the broken one has a 5 prime phosphate. The full length ones will have a G cap. Then we treat it with acid pyrophosphatase to remove the G cap, such as shown in here. So this part will be removed. So the mRNA will be left with a 5 prime phosphate. Then we are adding an oligo using RNA ligase. So RNA ligase will add this oligo to the full length one because it has a phosphate group, whereas the truncated mRNAs do not have a phosphate group anymore. So they don't get the oligo cap. Why didn't they have this? Because we treated with as alkaline phosphatase here, and this is where it lost it. Okay. And now you add oligo T, and both these can be will be amplified. Sorry, the first cDNA strand of full length will be made, and first cDNA of second one is also made. Okay, this truncated one. Sorry. But the point is, when we are synthesizing the second cDNA strand, we know the sequence of the oligo that we have added. So we use that sequence to synthesize the second cDNA strand. The truncated one does not have this oligo cap, because of which it will not be, the second strand of this one will not be synthesized. Okay. Yes, I'll stop here, and I would like to take any questions if you have. Quickly. Any questions? You may also type if you have. So there are 5 protocols. Okay. This is the start one. You don't have to. You should know the principle of mRNA isolation using oligo DT column and so on. The second one is, this is what you should learn using S1 nuclease. That is it is an old method of doing it, and homopolymer tailing. There are 2 problems with this. One is loss of information here on the 5 here, and the second one is directional cloning. So we need to, one of the methods is solving the directional cloning. Here they use it nicely because they're adding the linker prior to cleavage with S1 nuclease. That's a smart move. And then adding the second one for directional cloning. It solves the problem of directional cloning, but it does not solve the problem of loss of information. In the improved method, it is using this one. Oh, 1 second. This is using, we don't want to use S1 nuclease. So here what they use is by adding a oligo C. You can now, because I know that it is oligo C on this side, I don't have to use S1 nuclease. Instead, I can use a oligo G as a primer to synthesize the second cDNA strand. It's a good strategy, but it solves the problem of not to use S1 nuclease, but directional cloning is not yet solved. So in this one, this is using random primers for second cDNA strand and directional cloning also. Majority of the cases, you do get full length of the second cDNA strand. And yeah, there are 2 problems with most of the time libraries. One is incomplete reverse transcription by RNA polymerase. That is one problem. Second problem is broken mRNAs also get into make the clones. So we should be able to get rid of these things. For that, we have capture method, and the other method is called as oligo capping method. Capture method is to get rid of any sequences that are incompletely transcribed, like in here. By using RNAs, we are cutting it off, so they will now we will purify those that have the cap. Those that have the cap will not be acted upon by RNAs because they are full length. There is no single-stranded RNA available for it to act upon. Then we can use EIF4E affinity column to separate the capped ones from the uncapped ones. So these are eliminated from the cloning process or from the library. The last one is the oligo capping. Here the objective is to get rid of broken mRNAs. Broken mRNAs will not have GCAP. Instead, they would have had the phosphate, 5' phosphate. So treatment with alkaline phosphatase will get rid of the phosphate here. It will result in OH, whereas the cap, a full length one, will not be acted upon because it's a GCAP. Then we treat it with acid pyrophosphatase. Acid pyrophosphatase will cleave the GCAP somewhere here after these 2 phosphates, and it will result in a phosphate group. Then we add a oligo cap, which is a sequence, known sequence, and it will be added to the full length ones and not to these, because not to the broken ones or truncated ones, because it's a OH. Then because this upon adding oligo DT, both full length and truncated ones will get first cDNA strand. But for the synthesis of, during the synthesis of second cDNA strand, we are going to use a primer that is specific to the oligo cap that we added. So the second cDNA strand for this is synthesized, but for this, for the truncated one, it is not synthesized. Okay. I'll stop here. If you have any questions, please ask. Hello. Okay, I will post this today, and you can check it out and let me know if you have any questions.